Welcome to The Explainer. Today we're diving into a fascinating medical mystery. We're looking at a highly adaptable pathogen that, believe it or not, is probably sitting harmlessly inside many of us right now, completely unnoticed. But, as you'll soon see, its history? Yeah, it's anything but quiet. So, the central question driving our journey today is this. How exactly did a seemingly harmless bacteria, one that historically only bothered dairy cows, suddenly flip a biological switch and become a deadly, opportunistic threat to us humans? Part 1. From cows to humans, how a veterinary nuisance jumps species.
To understand this massive jump, we got to start with the very humble origins of our microscopic subject. The organism we're tracking is Streptococcus agalactiae. Now, that species name roughly translates to no milk, which actually reflects its historical identity as a cause of mastitis in cows' udders. Back in the 1930s, this pioneering microbiologist, Rebecca Lansfield, assigned it the letter B for one simple reason: bovine. I mean, literally no one thought it had any relevance to human disease whatsoever.
And this highlights a massive blind spot in early medicine. We were all familiar with Group A strep. That's the kind that actively makes us sick with things like strep throat. But Group B? It completely operates under the radar. It quietly colonizes the human body, usually just sitting there, totally harmless, as a normal member of our bacterial flora. No vaginitis, no gastroenteritis, nothing.
But the timeline of this evolution will give you absolute whiplash. For decades, this was strictly a veterinary issue. Then, suddenly, in the 1970s, there was this terrifying emergence of newborn babies suffering from sepsis and meningitis. The bacteria was so totally foreign to human medicine at the time that lab workers literally had to dust off veterinary texts just to identify it. It went from completely not existent in humans to a massive infant threat in a single decade, well before we finally developed life-saving medical protocols in the late 90s.
Moving to part 2, the ultimate opportunistic pathogen, a master infiltrator.
So to understand how this unprecedented jump even happened, we have to look at its biology. You see, bacteria exchange DNA all the time, and Group B Strep acquired new genetic material that allowed it to actually adhere to human cells. But here's where the historical context gets absolutely fascinating. This jump perfectly coincided with the widespread use of oral contraceptives in the 1970s. Now, while it's still being studied, researchers strongly suspect that the resulting hormonal changes altered the surface proteins of human cells, basically giving Group B Strep the perfect new environment to just latch onto. Crazy, right?
And once it attaches, man, it does not let go. Group B Strep deploys highly adaptable attachment proteins to grip onto specific substances in damaged human tissue. We're talking things like fibrinogen, keratin, plasminogen, and laminin. These are basically the scaffolding of healing wounds. This vice-like grip makes it incredibly dangerous for folks with conditions like diabetes, allowing the bacteria to form a biofilm and cause a chronic festering infection that can be completely refractory to treatment.
It has this truly elegant, albeit completely destructive, biological toolkit. For instance, it uses a weapon called hemolysin to literally destroy our immune defenders right on contact, just wiping out mast cells, macrophages, and neutrophils. Then it builds those protective biofilms I mentioned to keep a low profile and evade detection entirely. And incredibly, it uses chemical signals to switch its own adaptable genes on and off. It basically adjusts its attachment molecules perfectly to whatever environment it happens to find itself in.
Part 3, the threat to newborns, exploiting vulnerability.
Pivoting back to that terrifying outbreak in the 70s, we can see exactly why infants are so uniquely susceptible. At birth, a baby's intestinal tract is entirely sterile. So if they happen to swallow material from a carrier mother in the birth canal during vaginal delivery, the organism gets right into the gut. And because a newborn's cellular tight junctions, those microscopic seals between cells, aren't fully formed yet, the bacteria just slips right through. It enters the bloodstream, completely bypassing their underdeveloped immune system to infect the meninges and cause life-threatening meningitis.
Part 4, a life-saving medical solution, the 1990s protocol.
So without a vaccine available, medical science had to get pretty creative to stop this thing before it even begins. The magic number in their protocol? 3. Exactly 3 weeks before an expected due date, an expectant mother is swabbed and cultured for Group B Strep. By identifying carriers early, doctors can prepare a highly targeted defense for the exact moment of birth.
And here is the absolute genius of this protocol.
They don't actually try to clear the organism from the mother ahead of time. Instead, if she harbors it, intravenous antibiotics like ampicillin are administered right during delivery. Why? Because the antibiotic immediately enters the baby's circulation. It literally pre-arms the newborn's bloodstream. It completely wipes out the invading bacteria before an infection can even take root. I mean, this single protocol caused newborn sepsis rates to just plummet in the late 90s.
Which brings us to part 5, the rising antibiotic threat, an ongoing arms race.
Now that protocol was a huge victory, but we are essentially locked in a biological arms race here. While penicillin and ampicillin still work today, consider the alarming reality of our alternative defenses. In some strains, an astonishing 80% are now highly resistant to tetracyclines. And about 40%, they just laugh off alternatives like erythromycin, azithromycin, and clindamycin. Our chemical arsenal is visibly shrinking right before our eyes.
And as if antibiotic resistance wasn't bad enough, it's actually evolving new weapons. Certain strains of group B strep cannot produce a dangerous exotoxin that acts as a superantigen. This essentially short-circuits and overloads the human immune system, which can sometimes result in sudden, massive toxic shock syndrome. It's a pathogen constantly looking for new ways to wreak havoc.
This quote directly from the sources really hits the nail on the head. Group B strep is the ultimate opportunistic pathogen. Think about it. It transitioned all the way from a harmless bovine nuisance in the 1930s to a master human infiltrator. For most of us, it just sits quietly as part of our normal flora, totally unnoticed. But given the exact right circumstances, like an unhealed diabetic wound, or the extreme vulnerability of a newborn, it strikes with deadly efficiency.
Since this bacteria is constantly acquiring new genes, adapting its armor, and building resistance to our medicines, it leaves us with one pretty chilling question. As its antibiotic resistance grows, what exactly is the next chapter in this opportunistic pathogen's evolutionary journey? Are we prepared for its next major mutation? Or will Group B strep catch us completely off guard once again? Something to think about. Thanks for joining me on today's Explainer.